So that area, okay, so what we're now going to talk about is how we do this process. I've explained why we're doing it for those two reasons, for the bulk production of proteins like human hormones or to genetically modify plants. We don't do it yet to genetically modify animals. There's no bacteria that injects plasmid into animal cells, so it really is a plant genetic modification. That's why other techniques are being investigated as to what could be used for animal cells. So it's purely plant reasons this time. Okay, so what we're going to talk about first of all is how we get our target gene, insulin gene, I'll take that example, into a plasmid. This is called recombination. And so what we're doing is, as I say, we are joining human DNA in our example, and that's the insulin gene, and plasmid DNA. And recombination again is the joining of DNA from two different sources or species. Okay? So we've got plasmids and we've got human DNA. They're the two different ones, and we're going to join them together. Okay. So here's a flow chart of the story. I just need to take that down a bit. Here's a flow chart of how the story goes. So start off with the right-hand side. This is our example. This is our human DNA. This is the DNA that we extracted from human cells. We then added the restriction enzyme that would cut the DNA at the correct positions so that we could isolate our target gene. And we've run it, we've run it on a PCR and got lots of copies of it. We've done the gel electrophoresis. So here we have our target gene. So it's going to be isolate by the gel, and we're going to PCR it. So we've got lots of copies. So that's the story we've talked about already. Now, here on the left-hand side of our picture is our plasmid, our circular piece of DNA. It's a complete circle. Remember, I did say at one part, and I'm just colouring this part, this is where we've got that resistance gene that we select. And so the scientists will select a plasmid that has a particular resistant gene. Our example, our gene for resistance is going to be against the antibiotic ampicillin. So we've got an ampicillin-resistant plasmid, if you like. So it's a complete circle. Now, we have got our insulin gene, our target gene. We've got our plasmid. If I just mix them together, they will stay individually as our insulin gene and our plasmid. They need to be joined together. The bacteria needs to have the plasmid as a complete circle to take it up. So we have to get our gene into the circle. I could just attach it to the edge, but the bacteria won't take it up like that. It has to be incorporated or included in the circle. So in order to do that, what the scientists do is they cut the plasmid. They cut the plasmid with a restriction enzyme. Plasmids are DNA. But they don't just use any restriction enzyme. They use the same one that was used to cut the target gene. And this is so they will have complementary ends. Because we're using the same restriction enzyme, it means it's going to cut at the same sequence, and therefore when it's cut, the sticky ends in this example will match what the sticky ends are on our human DNA, and our example is insulin. And these ends here on our human DNA will match the ends on the cut plasmid. So by using the same restriction enzyme, we're going to be able to create these matching ends. So now I have my plasmid cut. I haven't taken a piece out. I've literally just cut it and the plasmid opens up. And I have my target gene, my insulin gene, and we just mix them together. So we're going to mix the plasmid, which has been cut, and the target gene, which is our insulin gene. And as you can see, the complementary ends will join. Because they match. And so you can see in these regions here, the matching of the nucleotides. So they will come together. But they won't stay together. So what we have to do is we have to make sure this joining is strong and held tight. This is where we use an enzyme, which is the third of our enzymes that we had in our list at the start. It's called DNA Ligase. Ligase means to ligature. The word ligature means to join. And so what this enzyme does, it joins DNA. And so it's going to join the target insulin gene DNA to the plasmid DNA. So what we now have are very strong bonding of the plasmid and the target gene. So we're going to have these really strong bonds so that when the plasmid gets into the bacteria, it won't break apart. It will be nice and complete. What we have created here, this is called a recombinant DNA plasmid. This is recombinant technology, creating a recombined plasmid with target gene. So now this recombinant plasmid isn't just like the original, it's got our gene in it as well. So therefore, if we can get this plasmid into bacteria, yes, the bacteria will have the plasmid, but it'll have our gene too. So this is step one, is creating the recombinant plasmid. That's the first step in our process. Step two in the process is getting the plasmid into the bacteria. And that is called transformation. So recombination is making the plasmid with the gene in it. Transformation is now changing the bacteria, making the bacteria take up the plasmid.